Unit 5, Section 1, Serology. Okay, serology is the analysis and study of body fluids. This is not just the study of blood, but also saliva, semen, and urine. Large component of this is going to be DNA. Since the late 1980s, serology has become a much larger field of study. And as we develop DNA technology, that's also become much more integrated. A lot of crime labs actually are underfunded. We've talked about this before. And so a lot of crime labs actually don't have the money to do a lot of the DNA testing. So they'll have to send that out. But in the meantime, they'll use a lot of basic techniques that we're going to be looking at here. All right, so how is blood... what is blood made up of? It's made up of 4 major components here. The first is plasma. Plasma is the fluid portion of the blood. This is kind of like the fluid that kind of holds everything else. Okay, so it's made up mostly of water. Plasma is primarily water. And that's over half of blood is this component. And then there are different types of cells. There are 3 different types of cells we'll look at that make up the remaining portion. Erythrocytes, which are going to be your red blood cells. You have the leukocytes, which are going to be your white blood cells. And then you have your thrombocytes, which are your platelets. Each one has different jobs. Erythrocytes or red blood cells are responsible for moving oxygen through the body, also bringing waste products like carbon dioxide. Leukocytes are the white blood cells. Those are the defense against foreign invaders, against pathogens and disease and viruses and things like that. And the thrombocytes are the platelets. These are responsible for causing scabs and blood clotting. And then it says serum is the liquid that separates from blood when a clot is formed. And so a lot of times we'll talk about pulling out serum, which is going to be primarily plasma, but then also it's going to have some platelets and white blood cells in there as well. Just a quick look back, around 1900, Carl Landsteiner discovered that there are 4 basic types of human blood, so we're going to be talking about blood typing as well. And then later we discovered something called the RH factor, so that was in 1940. So you have 2 major components in blood. You have, when we do blood typing, you're going to have a blood type, and then you're going to have an RH factor. So for example, A positive, you'll have, maybe O negative, okay, AB positive. So that's how that works. So, couple basic terms to know. When we talk about blood groups, there is an A group, there is a B blood type, and then there's something called an O blood type, which has none of the antigens, or you could have both A, B together. So just an understanding of those basic groupings of blood. RH factor, which is that rhesus factor from the previous slide, it may or may not be present, so it's a plus or a minus on the presence of that. And then knowing the difference here between an antigen and an antibody. Okay, an antigen is a substance that can stimulate the body to make antibodies. So an antigen is a stimulant, or this is a signal. Okay. And then an antibody is the substance that reacts with an antigen. So we say here certain antigens or proteins are found in the plasma of the red blood cell's membrane, and that's what accounts for blood type. So to make that kind of full circle there. So we have a signal, and then we have a response, an antibody response. And then agglutination is the clumping or clotting of red blood cells. When you do your blood typing, you'll be mixing different antigens together, and when they agglutinate, there's another way to say when they clot. And then finally, we are going to be talking about the case of Dr. Sam Shepard. which is there's a Harrison Ford movie called The Fugitive, which is about Sam Shepard's story. Paul Kirk is just a big guy that was involved in the process into that case and looking at the blood stains, the blood spatters. Those were actually really critical components. So the video is super interesting. We're going to be watching that here in the next couple of days. So there you go.